So we know now about primary structure, of course, the sequence of amino acids. We know a little bit about secondary structures, smaller localized regions of folding like the alpha helix and beta sheet. And then we started looking at tertiary structures, which are much larger structures where you have regions of secondary structure kind of coming together in a more complex way. And I want to focus on tertiary structure a little bit because there's some really interesting properties in that regard. And then I want to talk about quaternary structure, which are when multiple proteins come together to make larger protein complexes, and some of the properties of those larger complexes. And something called allosteryS, which has to do with conformational changes, but we'll get into those details. So with regard to protein tertiary structure, the tertiary structures give rise to what are called protein domains. And as it turns out, most proteins have what are called a modular structure. That is, they have multiple functions. Usually proteins don't have one function. So for example, if you have a DNA binding protein that, let's say, is a polymerase, a DNA polymerase that replicates DNA, well, that protein actually has multiple activities. It has a DNA binding activity, and then it has the DNA polymerization activity. So there's two activities there, and some proteins have many more than that. But the finding is that these specific activities are independent and they're localized within specific tertiary structures. So proteins look like they are modular in their construction. So let me give you an example of what I'm talking about. So we're talking about protein domains, and the way I define it is distinct structural tertiary structures, protein structures with very distinct functions. And again, proteins often have multiple activities, and those activities are contained within distinct modules, and that's what we call a domain. So let me give you an example. This is p53. This is a protein that we've been working on for a long time, and I'll talk more about it later. It's a protein that induces apoptosis, a tumor suppressor. But the point here is that the p53 functions as a dimer. That is, two proteins stuck together. That's a quaternary structure, actually held together by amphipathic alpha helices. And so you have these two proteins stuck together in order to make the functional activity. And what it says below is that after it dimerizes, then it binds to DNA and it turns on genes, activates gene expression. Now, if you take that protein, if you take one of those two parts of the protein and you stretch it out, like take away all the tertiary and secondary structure and just stretch it out, and that's what's shown on the right-hand side. There's the amino terminus and the carboxy terminus of the protein, and I have three rectangular boxes. Those rectangular boxes represent very specific activities. They also are specific domains. That is, each one of those folds up into a very specific structure and then carries out, let's start on the right-hand side, that region carries out the dimerization, allows the proteins to stick together. The middle section is what's called DNA binding domain. That folds up, binds to DNA. Then you have the gene activation domain, which, when folded correctly, can activate gene expression once it interacts with the DNA. Now, these function independently. So you can take, for example, the dimerization domain and take it off of the protein, remove it, and that will fold up into the proper structure and dimerize with other copies of itself. So independent of the rest of the protein. You can take the DNA binding domain, you can remove that DNA binding domain and put it on another protein, let's say albumin, which never binds to DNA, and now we find that the albumin will bind to DNA. And so these are structural and functionally independent regions of the protein that fold up correctly and then carry out their function independent of the other regions of the protein. You can have multiple mutations in the activation domain, but it has no effect on the structure of the DNA binding domain and the dimerization domain. Those will still function appropriately. Now, knowing this, what some have hypothesized is that these structurally and functionally independent regions of the proteins represent independent activities that over time join together in order to make a protein with a more complex activity. So that these domains actually may have been separate at one time and then joined together in order to make a DNA binding protein that would lead to gene activation. So let me give you an example of why we think that is probably true. So here's an example of a study that was done. So the hypothesis is that complex proteins with multiple structures are derived from individual modules that have joined together. So the example I'm giving you is a DNA binding domain, which is called a helix-loop-helix DNA binding domain. Now there's lots of different DNA binding domains out there. Many proteins bind to DNA. They don't all have the same DNA binding domain, but in this case, we're looking at 3 different proteins, and you'll see they all have the same DNA binding domain. So it's a helix-loop-helix, and I'm giving you an example of 3 different proteins. One is called a cap protein. This is a 350 amino acid protein. Another is called lac repressor, 360 amino acids. And another one is called lambda repressor, 240 amino acids. These are very different proteins, but they all bind to DNA. Now I have the illustration of the linear protein in green, the amino terminus carboxy terminus, and the DNA binding domain, the helix-loop-helix DNA binding domain, is represented by the rectangular hatched box on each protein. Now the finding is that if you look at these 3 different proteins, their amino acid sequences are completely different. There's no similarities among these DNA binding proteins in terms of their primary sequence, except for the helix-loop-helix DNA binding domain, which in all 3 cases are virtually identical. I'm not going to say 100% identical because there may be 1 or 2 amino acids that are changed, but they are, for all practical purposes, identical, even though the rest of the proteins are completely different. So there's 2 possible conclusions from something like this. One is that these 3 completely independent proteins evolved very specific structure to bind to DNA independently. Or that the DNA binding domain, the helix-loop-helix DNA binding domain, existed already and then was somehow placed onto these different proteins in different places in order to give them the ability to bind to DNA. And we think that it's the second which is true, that this DNA binding domain, the helix-loop-helix, arose once, turned out to be a very good structural and functional activity, and then through DNA recombination moved the sequences around onto other proteins and then allowed them to bind to DNA. You can think of it as a way to evolve something quickly, that rather than reinventing a DNA binding domain every single time or to use something that's in existence. So those are protein domains. They're more complex tertiary structures, and they represent independent functional units on proteins, and that proteins are modular in their structure and function. Okay, so I want to move on and talk a little bit about quaternary structure. So let's look at the next slide. So quaternary structure represents proteins with multiple protein subunits. It represents proteins that come together. Like I said, large protein molecules are often comprised of multiple protein subunits. And many of the proteins that we'll be looking at, the DNA polymerases, the transcription factors, as we mentioned earlier, the ATP synthase, obviously multiple proteins. So there are larger proteins made of multiple protein subunits, and it ranges from something like P53, which is just 2 subunits. Here it's showing examples of other proteins. There's a dimer of the cap protein. This was the protein that we just talked about with the helix-loop-helix domain, and a dimer formed by the interaction of two proteins. And then we have a tetramer. Here they're showing neuraminidase, and it's formed by 4 proteins coming together. Hemoglobin is another protein that has 4 subunits. RNA polymerase, which we'll talk about a lot later when we talk about transcription, has 10 to 12 subunits. The ATP synthase, which we talked about, is 27. And then there's some that are much, much larger, like actin, which is a part of the cytoskeleton, is made up of individual actin proteins, and there can be tens of thousands of proteins that link together. Microtubules, which we'll also talk a little bit about, actually probably in a little bit, a few minutes, and have tens of thousands of tubulin proteins linked together. Now, these proteins self-assemble, and they self-assemble on the basis of what are called complementary surfaces. Complementary surfaces. Sorry, not surfaces. Complementary surfaces. That means surfaces that are made to link together. So this slide shows a very simple way of explaining what we call self-assembly using complementary surfaces. So if we look at A, there's 2 free subunits, and you can see how they can link together because there's kind of a complementary bend in the protein to make a dimer. And if you look at B, for example, that may be something analogous to actin filaments, where you have a protein which has a little bulge on it, and that bulge fits into that little groove that's on the protein, and so you get these things linking together. So these are referred to as complementary surfaces, and it allows self-assembly. Now, many of these proteins, or I say most of these proteins that have multiple subunits, undergo this self-assembly. And for one thing, I want to say it's important to remember what this self-assembly represents. The complementary surfaces, if we're looking at this figure here, it's showing you as a little bump and a little groove in the protein. But what I want you to remember is that that's not what they are, although they may be those kind of shapes, but they are all based on amino acids, because here we're talking about proteins. And the amino acids have R groups. And so the same kind of interactions that lead to the 3-dimensional structure of proteins, all the interactions that take place between the R groups, is the same kind of interactions that are taking place here. Meaning that on the surface of the protein, there will be specific R groups, and on the surface of the protein that will be interacted with, the complementary surface really represents another series of R groups that are exposed on the surface, that when these things are brought together, then you have these interactions that take place. They could be ionic, it could be hydrogen bonding, they could be covalent if they're two cysteines. And so that's what we're talking about. We're talking about amino acid interactions, the very same kind of thing we've talked about before. Now the other, well, let me just show you a couple of examples. I'm going to show you examples, two very interesting examples of self-assembly. So here are just two examples that are quite remarkable. Self-assembly. Proteins that have complementary surfaces. So the first example is a virus called tobacco mosaic virus, TMV. This protein was studied this virus was studied for a long time. And what the virus is made of is a single copy of RNA, as an RNA genome, and then it's coated with exactly 2,130 copies of a coat protein. They're identical proteins. 2,130 identical protein copies and one copy of RNA. Now, if you take purified coat proteins, so in other words, if you take the virus and strip out the RNA, purify the individual proteins and have them in a test tube, and then you throw them together with a copy of RNA, with sufficient numbers of copies of RNA, what happens is these molecules self-assemble in the presence of the RNA into a functional virus that will infect cells. So you can take the proteins, take the RNA, throw it into water, and it builds itself into an active virus. This is a dramatic example of self-assembly. Another one, which is maybe even more dramatic, is the bacterial ribosome. Ribosomes are extremely complex. Of course, we know they carry out protein synthesis. And with the bacterial ribosome, there are 3 ribosomal RNAs and 55 different proteins. With the tobacco mosaic virus, they were identical proteins, but with the ribosome, there's 55 different proteins. Now, believe it or not, the same thing is true if you take the 55 individual proteins in individual test tubes and then mix them together in the presence of the 3 ribosomal RNAs in water, because water is important. The ribosome will self-assemble and be able to carry out protein synthesis. Now, I'm exaggerating a little bit because it can't really be done like that, but the way it is done, and basically the principle is the same, is there's 55 different proteins and you have to add one at a time. And so people figured out the order that needs to be added. So you put the ribosomal RNA in, then you put in the first ribosomal protein, it binds. Then you put in the second ribosomal protein, it binds to the right place. The third, 1, 4th, 5th, and so on, all the way to 55, and then you get yourself a functional ribosome. So, again, the same kind of forces that we talked about before, all the amino acids on the surface interacting with the complementary amino acids through the same kind of interactions, hydrogen bonding, ionic, covalent, and so on. Also interactions between peptide bonds. And what I want to stress at this point is that this complex self-assembly is also built into the primary structure of the protein. So it's not just the folding of the protein into its precise shape, but what also is present on that primary sequence is the location of the amino acids that are required for these interactions to make this quaternary structure. And of course, all this is encoded in the DNA. Now, as it turns out, this kind of self-assembly happens, but there are numerous diseases which are due to what we call inappropriate self-assembly. That there are numerous diseases that are due to proteins aggregating where they shouldn't be. And so let me just give you a few examples of this. And then we're going to talk about one very interesting example. So I can give you a whole table with, dozens of different diseases, which are due to inappropriate complexing or inappropriate quaternary structures, but here's just a few. Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS. These are all very serious and very drastic neurological diseases, which we know, cause people to lose their memory, cause people to lose control of their arms and legs and not able to walk, and eventually, sometimes even the heart will stop. And all these are due to the inappropriate quaternary structures that are formed. In Alzheimer's disease, there's what's called an amyloid beta protein, which self-assembles incorrectly. It makes these amyloid plaques. There's also another protein involved, which is called tau, which self-assembles inappropriately, forms these what they call tangles. In Parkinson's disease, there's another protein that makes these large complexes inappropriately. In Huntington's disease, same thing. In ALS, there's a number of proteins where you get these large complexes formed, and what they call neurofibrils and these other inclusion bodies, which are basically precipitated proteins which occur inside the cells. Now, notice that all these seriously affect brain function. What happens when you get these amyloid plaques, or you get these Lewy bodies, or you get the aggregated Huntington protein, what happens is the brain cells die. And so it kills the brain cells, and when those brain cells die, then they give off materials, they're like byproducts that kill neighboring cells. So basically you end up getting cell death in the brain. Now, why are so many of these related to brain function? And that is, let me take another. You can lose, let's say 5, 10, 15, 20% of your liver, and not have any effect at all. Basically, liver will repair. You can lose 50, 60, 80% of your kidney function, and you'll still be healthy. In fact, you can clearly lose half of your kidneys, because you can give a kidney, you can donate a kidney and still be fine. So you can lose 50% of your kidney and you're okay. You can't lose 50% of your brain. You can't even lose a few percent of your brain. So that's why the brain is so sensitive to these kinds of defects, that any even minor death of tissue in the brain causes severe repercussions. Now, I want to tell you another example of protein self-assembly, abnormal self-assembly, and I want to talk to you a little bit about what are called prion diseases, because this actually just turns out to be a really fascinating story and very interesting and medically significant. So here we're again talking about abnormal self-assembly of proteins, and the proteins that I'm talking about are called prions. And the inappropriate self-assembly of these prion proteins, these particles, create diseases, again, seriously affecting the brain. Now, there's a number of these diseases which are due to prions. The prions appear to be infectious, and you'll see why they look to be infectious. So there's a number of different diseases that are due to prion infection. And I have 4 listed here. There's what's called Kuru, and I'll talk about some of these in a little bit more detail. Kuru exists in humans. Scrapie exists in sheep. There's another one called bovine spongiform encephalopathy, which is otherwise known as mad cow disease, which I'm sure a number of people have heard of, and I'll talk about it in a minute. And then finally, there's another disease which is called Creutzfeldt-Jakob disease, which is, again, in humans. So we first became aware of prions with the Kuru. And the Kuru was discovered, this particular disease was discovered in a population of people in New Guinea by anthropologists in the mid 1950s, early 1960s. What happened is these anthropologists were going in to these villages in New Guinea and just looking at behavior, looking at societal structure, and they noticed in a few of the villages that there were a large number of people, particularly women, a large number of people which looked like they had Huntington's disease. They were falling. They couldn't walk straight. They had severe dementia and very, very unstable and extremely sick. And when they looked at, so they started studying this disease, and it looked, they couldn't figure out. It looked like it was inherited in some way because some families had more than others. But after a while, they found out that they realized that it wasn't. That it was really infection. It was due to an infection. As it turns out, and the reason why these people were getting this disease and spreading it through their community was that these people practiced, these people practiced cannibalism. Cannibalism, yep. And what was happening was they were eating the flesh and meat and muscle of people who died. Now, in many cases, people who died were dying from this particular disease. And in this culture, they felt it was honorable to eat the dead person, your family member, to eat the family member. And so they would divide up the portions, and the females got the last pick of the goodies. And so the females ended up, in many cases, eating what was left after the males had their share. And that often was the brain and the spinal column, some other muscle tissue, but the women were more susceptible and more prone to eat those parts. Well, as it turned out, these were due to prion diseases, and these prions infected the brain tissue in particular. And so when these women were eating the brain tissue, then they would later come down with the disease. And sometimes it took a few years. It could take as much as 30 years for it to appear. So, and then people, of course, started studying these prions and what they were. And we'll talk about them a little bit later. So it's discovered there, and then after that, people. By the way, cannibalism is outlawed in New Guinea, although some of the communities still practice it in hiding, and some of the people are still showing these diseases because they're still around. Some of them were children at the time in the 60s, and so now they're in their 50s, 60s, or 70s. Then there's scrapie, another one. Disease that comes in sheep. This may be some genetic component to that. Then there's the bovine spongiform encephalopathy, which is mad cow disease. So let me just tell you a little bit about mad cow disease. Mad cow disease, there was a large epidemic of mad cow disease, particularly in Great Britain, some parts of Europe like Belgium, maybe a little bit France. In the late 1980s. And what happens with this is that the cows basically behave like the people did. They lose their ability to walk. They're falling down all the time. They're kind of moving in circles. They're running into things. They can't walk. It's very sad to see. It's like a dementia in cows, and plus they lose their ability to walk and they're falling, and so that's referred to as mad cow disease because they look kind of mad, not mad angry, but mad insane. Well, what happened in England, or in Great Britain, was that it became apparent that there were a number of cows all over the country, all over the island basically, that were showing this disease. And the farmers, the ranchers, never reported it. And so by the time it was reported, there was an estimate of around 450,000 infected cattle went to market. Almost 500,000 infected animals were put into market, and the meat was processed and distributed to people. By the way, that's what led to the Creutzfeldt-Jakob disease, which we'll talk about in a minute. Once that was found out, this whole episode went from around 1980 to 2000. About 180,000 cattle died from the disease. But once it was made apparent, then the government went out and killed every single cow, cattle in Great Britain. And the estimates are that 4.5 million were killed to eliminate the disease. It completely devastated the meat market, obviously, in Great Britain and some other European countries. We never had it here in the United States. There have been a few isolated cases, but the FDA gets on it very quickly, and they kill the animal, they quarantine the rest of the herd, and then there's testing that can be done. And so they killed 4.5 million cattle in order to eliminate this. And in fact, I know people, and you also may know people who were in Great Britain at that time, and you can ask them, but I know people who grew up during that period, or were in England during that period, and they remember taking rides into the countryside and seeing these enormous bonfires going. And what those were the cattle being burned. Well, 450,000 went into market, were eaten, and of course we know from Kuru that if you eat infected meat, that you can come down with this disease. And as it turns out, there's around 200 people in England at this time. Maybe the numbers are a little bit higher. I'd have to look it up. But the last I looked up, there are around 200 individuals who have Creutzfeldt-Jakob disease due to eating the infected meat. In the U.S., there were 4 or 6 people that came down also with Creutzfeldt-Jakob disease. And if you look at their life and their travel patterns, all of them had been in Great Britain during that period of the epidemic. So this was a disaster. It was a huge public relations disaster, a huge scandal in Great Britain. And then above and beyond that, the question was, how did the cows get the mad cow disease to begin with? Where did it come from? So there was another secret that was being kept, and that was that although it was being publicized that the cows were eating vegetarian diets, in fact, they were not eating vegetarian diets. They were mixing the vegetarian feed with animal products, byproducts, and in particular, it was sheep. So they were using sheep byproducts and mixing it in with the cow feed. And so the thought is that some of the sheep that were put into the mix of the cow feed that was used all over the country was contaminated with scrapie, and that was the origin. So those are prion diseases. And so let me just tell you a little bit about what they are. They're referred to as infectious proteins, because if you ingest it, you end up getting the disease. So the prions are pure protein. There are 254 amino acids, and there's no known infectious agent that is only pure protein. Any infectious agent, like a virus, has a genome which encodes proteins like RNA genome or a DNA genome. So coronavirus, for example, has an RNA genome. Flu virus has an RNA genome. So it's not clear, it wasn't clear at all how these things would be infectious. So there's 254 amino acids, and as it turns out, our brains and the brains of sheep and other mammals have a protein which is a prion protein, but which is normal. It does not have this lethal kind of effect. So I have that written as PN, which means a normal prion protein. These are non-infectious. They occur in the brain, and they're thought to be involved in copper transport. So we make a protein that is this 254 amino acid protein, which is crucial for normal brain function. Now, in the normal one, that's 254 amino acids, and if you look at the structure from crystallography, it's all alpha helices, and they exist as a monomer, single proteins in the brain. If you look at the infectious prion, the one that causes disease, it's also 254 amino acids, and in fact, it's the same exact sequence. 254 amino acids, they're identical between the normal and the infectious one. But for some reason, the infectious 254 amino acid prion forms beta sheet structures. Not the alpha helix, the beta sheet. And we know these beta sheets can kind of line up and form these aggregates, and that's what happens. Now, why would the same 254 amino acids do two different things? We don't really know that. It could be that the 254 amino acid protein in the infectious form may be modified in some way after synthesis. Maybe the addition of some other element or some other small protein gets attached to it, causes it to aggregate. So the ones that cause disease aggregate just like with the Alzheimer's, and that's what causes the lesions in the brain. So here's the next slide, shows the model of how these things are thought to quote-unquote replicate. So here's the model, and this is pretty much what we believe happens, a model for replication of the prions and the disease. If you look at the left-hand side, that's what occurs in a normal brain. You get these normal proteins being formed. They form alpha helix, and they exist as single proteins in the brain cell, and presumably do something with copper. In the diseased tissue, you have the prions, but the prions are infectious. They form beta sheets, and you see they undergo this self-assembly. So what happens is, if you ingest diseased tissue, you are ingesting the infectious prion, or many infectious prions, and the infectious prions have the ability to convert the normal forms into a prion-like beta sheet form. So they force the structure to change. So what I've shown in the middle is once you have the diseased prion entering the brain, and the body in your brain is producing the normal prions, once they bind together, it forces the alpha helix into a beta sheet form. And then, of course, those aggregate, and then they cause cell death in the brain. So what's happening is they're not replicating. It looks like they're replicating because you get more and more and more of the defective prions. but they're not replicating. What's happening is that our normal brain cells are providing the substrate in order for the recruitment to occur. So we're producing it, it gets recruited, converted, and then you get the aggregation and the cell death. Okay, so that's pretty much what I wanted to say about protein aggregation and protein complexing. And I want to talk about another very important function of proteins, and that is their ability to undergo controlled changes in their structure, which will then control their function. And that's called allostery. So what I have written here is some proteins can undergo a regulated or induced conformational change. That can change their conformation a little bit. And that conformational change causes a change in activity. And that is referred to allostery. Sometimes it's referred to as an allosteric transition or allosteric regulation, where you can change the activity of, or not you, but the cell can change the activity of a protein by altering its function, by altering its structure. So at the bottom, I have just kind of a simple drawing, and there's also one on the right-hand side where I have this drawing. On the left, it shows a protein that's supposed to represent a protein with a little indentation on it. And then in the middle, there's a hatched rectangle, which is referred to as an allosteric regulator. So this is a compound. It could be a sugar, it could be another protein, it could be ATP, it could be GTP, it could be any one. It could be an amino acid. It depends on the protein, but it binds to that allosteric site on the protein, and that changes the structure. It causes a forced change in the structure. And so what I have shown on the right is it changes it to a square. You know, it's not really changing it to a square, but it's clear that the structures change ever so slightly. And that change could cause an inactive protein to become active. Or so you can turn the protein off and on by taking this thing off and on. Or it can make an active protein inactive. Or it can make an active protein more active. Or sometimes it can change its activity completely. This is an allosteric regulation, and it shows on the right-hand side, you know, a similar kind of thing. Here it's showing just some protein that binds sugar. You know, it's like glucose. And it's showing that it's inactive on the top, but it's activated by ADP. And when the ADP binds, it causes an allosteric conformational change that allows it to bind sugar. So it becomes active. So that's an allosteric regulation. Now, there's movement that's going on here. I'm showing kind of a change in structure, and this is showing opening and closing. You know, these are all representations, but in fact, there's movement that goes on. Let me give you another example in the next slide. So this is an example of a protein called multi-complex protein called aspartase transcarbamylase. You don't have to know much about this protein. You don't really have to know about what it does, but it is involved in the biosynthetic pathway of some of the nucleotides. But that's not really what's important here. What's important here is that it's regulated through an allosteric change in its structure by CTP, which is one of the nucleotides, cytosine triphosphate. And what's shown is an active enzyme on the left-hand side. It's active, so it'll be involved in synthesizing nucleotides. And then there's a regulatory site, which is an allosteric regulatory site, bound by 4 molecules of CTP. You can see them on either side, and that causes a allosteric regulatory change. And you can see they represent some kind of change in the structure of the protein, and it turns it off. Maybe since this thing is involved in nucleotide biosynthesis, if there's enough CTP in the cell, then you don't need to make more CTP. So in a sense, it's telling it, okay, well, you can turn the enzyme off. We have enough. So that's an example of binding to a particular compound, CTP. But the allostery can also be induced by modifying the any amino acid by what's called phosphorylation. Let me show you that in the next slide. So we'll talk more about phosphorylation much later in the semester when we talk about signaling and signal transduction, but phosphorylation is an extremely important way of inducing allosteric regulation changes, and which can cause a drastic change in the activity of a protein. So we look at the bottom. Let's not look at the top for a second. If you look at the bottom, it shows a protein off, and then if you have what's called a kinase is a enzyme that takes one of the last phosphate off of ATP and links it to an amino acid, and in this case shows it turning it on. It can be the reverse, that is, you can have a protein that's on or active, and then you can have the kinase activity activated, takes the phosphate off ATP, links it to the protein, and makes it inactive, turns the protein off. And then there are proteins which are called, other enzymes which are called phosphatases, which can remove the phosphate and then again turn the protein off or turn it on depending on what protein you're looking at. So kinases and phosphatases are important. And it shows up at the top. This usually happens. This happens on R groups of amino acids that have an OH. So serine is one, tyrosine is another one that are the most common. And so what it's showing is on the left-hand side, you see the CH2OH. That is the R group of serine. And then the protein kinase comes in, uses ATP, and then adds that phosphate, as you can see on the right, onto the oxygen. And so it changes the charge and the structure of that amino acid. And of course, that can completely change the overall structure of the protein. This could be something, for example, that happens to the prion proteins. You can get some kind of phosphorylation on an amino acid that can then change the structure. So phosphorylation, an important regulatory event. Now, these things move. Okay, the structures change. And I just want to show you just one simple example. Not a simple example, one example. So this is the structure of an enzyme called pyruvate kinase. Again, another kinase which is involved in activation, in phosphorylating other proteins. It's regulated by a allosteric regulator called phosphoenolpyruvate, which I'm showing in the middle, sometimes referred to as PEP. So this is the crystal structure of the pyruvate kinase without on the left and with the PEP. And you can see from the crystal structure that there's clearly a change in the conformation of the protein. Now these changes in conformation can be minor. It can be the rotation of a helix. It could be the movement of a beta sheet. It can be the loss of interactions between two essential amino acids that are required for the structure. So there's clearly movement that goes on. So allosteric changes represent movement. And so there's a whole class of proteins which are called molecular motors or motor proteins, which undergo movement based on these allosteric conformational changes, and that movement then is used to do work. So it's not just turning off or on, but it's actually used to do work. And in most cases, that work is movement. And I'll show you exactly what I mean by looking at these what are called motor proteins. So there are now huge families of motor proteins that have been discovered and studied. Sometimes they're called molecular machines or motor proteins. And they all carry out some kind of movement or transport. So let me give you a few examples of some of these. So on the left-hand side, I have the proteins. So kinesin, dynin, myosin, which is involved in muscle contraction, helicases, the sodium-potassium pump, which pumps ions, the ATP synthase, which we looked at when we looked at electron transport, and I mentioned a lot at that time that it was considered a molecular motor with moving parts. And those moving parts actually pump protons, and it's reversible. It's a reversible machine. But what's turned out to be really interesting are these motor proteins like kinesins and dynins, which cause the movement of vesicles. So on the right-hand side, I have the function. So kinesins are proteins that are involved in organelle transport or vesicle transport. Dynins are involved in protein or RNA transport. So organelle transport, that means moving a mitochondria from one part of the cell to the next, or moving a chloroplast from one part of the cell to the next, or vesicle. The dynins are involved in moving proteins or RNA transport. That is, with stuff that happens in the cell, we used to think that it was just like random diffusion. You know, how does the protein get to the nucleus? Diffusion, Brownian motion or something. Some of that may be involved, but what we're learning now is no, there's no such thing as randomness, really, that the proteins are moved specifically by these motor proteins to where they need to be. If you look at a cross-section, or if you look at a plant cell under a microscope, you may have done this in Biology 101, and what you see is the chloroplasts moving around the cell. They're moving around the cell pretty quickly. And they call that, it's called cytoplasmic streaming. But the cytoplasmic streaming is movement by these motors. So let me just tell you a little bit about kinesin. Okay, we're almost done. But I want to tell you a little bit about kinesin. Kinesin is a motor protein involved in organelle transport. And here's the protein at the bottom. There's two heavy chains, which are quite large, 1,200 amino acids. Two light chains. They're also reasonably large, 600 amino acids. And if you look at the protein diagram below, the heavy chains have the amphipathic alpha helix that cause these things to wind, just like we saw with the myosin a couple days ago. So you get that length of protein. On the left-hand side is what's called the motor domain. That motor domain has a number of activities. It binds and hydrolyzes ATP. And it also binds to microtubules. And it moves. And if you look at the right-hand side, it's what's called a cargo binding domain. That's where the light chains are. And the light chains, the different light chains that exist, lead to the specificity in which kind of cargo would be binding. So it's a cargo binding domain. In this case, we have it binding to mitochondria. So these things move organelles around the cell. And here's kind of an illustration of how it works. Okay. So this represents the movement of kinesin. It moves along a microtubule and carries an organelle or cargo like a vesicle or in this case a mitochondria. Microtubules, we mentioned, are long polymers of the protein tubulin. They're part of the cytoskeleton, and kinesin binds to the microtubules. So I'll just walk you through it. So at the very top, on the right-hand side, it shows the kinesin binding through the tubulin binding domain, and on the right-hand side is binding to the mitochondria. Now, when ATP binds, and everything I'm telling you here is happening through allosteric conformational changes. So the binding of ATP causes an allosteric conformational change that allows the lower part, the lower leg, let's say, to come off of the microtubule and move over a little bit. Then the hydrolysis of the ATPs, you can see the ADP plus the phosphate. There's a hydrolysis that takes place, taking that last phosphate off, but it's still in the complex. That causes the leg, which has moved, to bind back down to the microtubule. Then the next step, the release of the phosphate, causes an allosteric conformational change in the upper leg, and it comes off the tubulin and moves over a little bit. And then the release of the ADP causes the upper leg to bind back down to the microtubule. So it took a step. So these things take steps and they move down microtubules. They carry cargo. And people have done work to study these things and to measure the speed. You can measure the speed of how these things work or how these things move by labeling, let's say, the kinesin or the mitochondria with a fluorescent dye. And then looking under the microscope, you can watch it move down a microtubule and you can measure the rate. And it's measured the speed of about 3 micrometers per second. And so I translate that into something that makes more sense to us. It can go 10 inches per day. So they can move 10 inches per day, which is phenomenal, I think. And then if you look at a cell, if an average cell might be, cells are different sizes, but let's say some average cell is 25 micrometers across, then it only takes 8 seconds for a kinesin molecule to move a mitochondria from one side of the cell to the next. So if the mitochondria are needed at a particular side of the cell, they will start moving them down by this transport. Okay, so that's transport by kinesins, and basically all built on allosteric conformational changes. Okay, we've gone for almost an hour, and we'll stop here. And I will say next time we still have a little bit more to talk about. I'm going to talk about enzymes because proteins can be enzymes. Okay, we'll talk about that next.